Thank you for joining today's panel discussing AI in drug discovery. My name is Tim Hohm. I'm the Director of Commercial Strategy and Business Development at Optibrium. We have a great panel assembled today of seasoned drug discovery executives who will provide you with their view and experience with AI in the area and to what extent AI has managed to deliver on the promise of transforming drug discovery. We will have Jeff Blaney, who is Senior Director of Discovery Chemistry at Genentech and leads the AI machine learning efforts for small molecule preclinical drug discovery. Then we have Darren Green, who's the head of cheminformatics and data sciences at, I think, Darren, as of yesterday, you're officially GSK. Then we have Julian Lever. Julian is head of discovery at New Equilibrium Biosciences, a young biotech company. The final panelist we have is Matthew Siegel. He's CEO of Optibrium. With that, let's jump into the panel. And Jeff, you're leading Genentech's AI ML efforts for small molecule discovery. So where do you see AI today, and what are notable successes in the area?
Yeah, first off, I think what we're attempting to do in small molecule or large molecule drug discovery is relatively different than many other areas where we're being asked to invent and discover. But if you think about it pretty carefully, there are very few reported examples where AI has achieved this. AlphaGo is one of the most stunning. I love that cover photo from the New York Times of Lee Sedol, the world's best Go player. The picture tells it all, right? And what DeepMind was truly impressive. However, they had a big advantage over us. You know the rules of the game, and you know how to assess victory. Designing something new means you either have to know both of those, or you've got to have a large amount of data. Small molecules is generally a small data, not a large data regime. And none of us, including people who put many drugs on the market, can look at the famous chart of the top 200 selling drugs and say why one is a drug and another close analog is not. We can't assess victory very well. So we don't know the rules. We don't even know how to measure victory. However, there are clear opportunities. And as I was given the opportunity about 2 years ago to start building a strategy at Genentech for how to invest a number of new hires to broaden our efforts in small molecule machine learning. We focused on these areas so that the black ones are well established, not just at Genentech, but across the industry. They've worked really well with at least 15 to 20 years experience, especially in DMPK ADME and predicting physical chemical properties. The reasons for that are relatively clear. We have large data there. We measure those experimental endpoints on the majority of molecules we synthesize because they're not specific to a target. We care about them for all of the molecules we work on. The early models were support vector machines, random forests, multivariate Q-star. There's a variety of them that all work pretty well, and certainly well enough to provide a probabilistic model to impact which molecule we should synthesize next. So far, I've seen only incremental improvement from state-of-the-art deep learning methods in these areas over more classic methods. Some early signs where transfer learning approaches help. Most of the methods don't give any mechanistic insight, but still they can be useful to provide guidance of which molecules are a better bet. I've cited just a couple, I think, nice examples from the literature of multitask approaches to predict potency and properties. Nice work from Novartis, also from Matt and his colleagues at Optibrium. I imagine others will talk more about that. But I think these areas that are in the black text have already been well proven to work, and I would argue have had the most impact in design and choosing which molecules to make next. The green areas are newer, but also I think are working really well. We chose not to invest in building our own tools for retrosynthetic analysis because we could see that many external tools were coming. We're using a couple of the ones I mentioned on that slide with pretty positive reviews. Again, retrosynthetic analysis, you have very large corpus of reaction data, and victory is defined. When a chemist draws a molecule into Marvin or ChemDraw, it says, make me that. That's victory. Quantum mechanics, Adrian Reutberg published some very nice work called ANI a few years ago. I think that's now sort of a classic hybrid physics-based and ML approach, where you use a data set trained on high-level quantum mechanics in this case to train machine learning models to predict those results. And these are ones that I think are exciting, but less well proven so far. The first one comes out of Schrodinger's work. This is an example similar to the quantum mechanics one. In this case, you use another physics-based computation-intensive method, free energy perturbation, to build a data set of specific molecules that you're interested in to train a machine learning model so you can run at much larger scale. And I think the work published from collaboration between Google and XChem in 2020 and JMed Chem, super exciting. And this was using experimental DEL, DNA encoded library, screening to train a binary classifier machine learning model, which they then applied to virtual screen of enamine molecules to predict actives with a higher than expected success rate. Some of those molecules relatively novel with respect to the training set. Many groups have invested in sort of cloning this approach. I think that's super exciting because to me it gets to the crux problem. In this case, you get an experimentally trained scoring function. There was this really exciting paper in early 2020 from Stokes et al. This was Regina Barzolet's group and others on a machine learning model trained on phenotypic screening data that worked quite well. We're seeing that play out in other areas as well. Something that doesn't get quite as much attention, but I think is also really important. From the philosophy, let's go where the problems make sense scientifically for machine learning to impact. In analytical and purification chemistry, machine learning methods that can learn from large amounts of data we have so that the experimentalists in those labs do not have to do as many empirical experiments to choose what's the best way to purify a new molecule. Out of all the purification methods, where should they start? Faster ways, for example, to measure concentration. And then in process chemistry, lots of work in academia, and internally we're doing this as well, not to design molecules, but to predict optimal reaction conditions for synthesizing them and scaling them up.
Darren, I know GSK is quite active in the AI area as well. So could you share a couple of activities you engaged with and what type of value they have created for the organization?
Yes, certainly. So I think I'll call out molecule generation and ideation as something that machine learning is really helping with. We actually combine classical sort of cheminformatics methods with machine learning for this. Because we're a big pharma company, we have lots and lots of chemistry data. I can go back decades of hundreds of medicinal chemists and see what they made and see what the effects of those changes were. And so that's actually a very powerful way of reusing that information to generate new molecules. And actually, we published a paper where we passed the Turing test with our chemists. The machine generated, what would you make from here? And the machine generated things, and the chemists couldn't tell whether it was another chemist or the machine that had generated those ideas. So these are pretty powerful. And then as you get more data, you can start bringing in the machine learning methods, recursive neural networks and so on, which allow you to use the SAR you've generated and do transfer learning onto a pre-trained model. And we find them quite complementary. So we're really using those an awful lot now. And it's a for a computational chemist is a very, very powerful thing, changes the game, because all of a sudden you can generate 100,000 ideas, which are all plausible and which are all interesting. And then it comes down to what Jeff was saying, is like, okay, how good's my scoring function to select what are the really interesting molecules? One interesting fact, I think, from our pilot projects is we looked at the synthetic productivity of the chemists that we were making compounds designed with the automated design methods, and we could see that actually they were at least as productive as they were making their own molecules. So there is some inherent synthesizability if you're reusing data. There's something there, even if you haven't got the mechanistic stuff. There's something there that means they're synthetically plausible. I'll agree with Jeff. QSAR is very well established, and we've just completed a very large rebuild of all of our DMPK models, and we've used sort of modern technology and multitask learning and all these good things. And for some models we have seen a significant impact. We've got some strikingly good models, we think, now in some endpoints there, such that we're now looking at, should we, for some projects, can you actually replace the assay in the screening cascade because the model's getting that good? So that's very interesting. The other thing we've seen as a side effect of the lot of noise around AI, ML, and in the community, seeing a lot greater use of what we would term local models for projects. So you may have a global model built on all your data, but for some chemical series, it just doesn't work, and the demand to quickly get a local model. And for me, that's a sign of sort of uptake and people really buying into the idea that these predictive models have value. And so we typically are doing a lot more of that. And finally, the thing I'd say is a real quick win is something that's hard, that humans can't do very well, and solves a problem. So, and it's a bit outside of chemistry, but image analytics. So, if you're doing cell painting or you're doing some of these very interesting high content experiments, extracting features out of images, humans are very slow at doing that, and it's very difficult for them. We're fortunate to have a very strong AI/ML group, central AI/ML group in the company that has a great deal of computer vision experience, and so that's been really impactful. Is enabling that kind of technology and bringing it into the organization.
That definitely sounds great and interesting, Darren. Julian, you're currently working with a smaller company, and what is your experience, and what would you say are the conceptual differences for smaller biotech compared to what Jeff and Darren have been sharing?
Yeah, I mean, from a historical perspective, the majority of the work that I've been involved in on AI was from Constellation, my previous company, because we were working on some programs there where we had some pretty challenging chemistry, which was outside of the normal chemical space, we would say. And so a lot of the models, a lot of the standard models didn't really work because none of these compounds were part of the training sets. So it's one of the main problems with a lot of this predictive modeling tools, right, is they interpolate very well, but they don't extrapolate fantastically. So trying to figure out how to handle that, we actually worked with Octibrium on some of the early versions prior to the Sorella platform in order to do exactly that and help build up some of our capabilities. The local models was pretty much the only options we have, of course, because when you're in a small company, you don't have access to 20 years of vast data sets, and a lot of the literature data sets that are available are kind of curated and limited as well. So in terms of what we did there, it was applied to our specific problems. Collaboration was key to try and access some of the cutting edge technology. I'll come on to maybe some of the challenges a little bit later on for the large versus small company, but in terms of the success here, I think it was successful for us in terms of how to handle the sparse data set problem, how to generate ideas based around your earlier ideas, extrapolation, at least marginal extrapolation based on your data sets. I think one of the most important values that I found from it, to be honest, as we just mentioned before, you can generate a lot of ideas now, and quite a lot of them look pretty feasible. And then from a synthetic perspective, you can figure out how to make these in a reasonable way. So which ones should you make versus which ones should you prioritize, or which ones should you not make? And that was one of the biggest values for me, actually, from a pure synthetic cost. It's expensive. Chemistry is expensive. So, you know, if you can not make 10% of the compounds that you were planning to make, that saves you a vast amount of money. It's not really an intuitive thing to think about, what do I not want to make? But it really was really helpful for us to be able to optimize our value add from our synthetic chemistry team that we had, both internal and external, at CROs, to be able to make the right compounds and move the compounds and the projects forward faster. And then the other thing that I thought was pretty interesting was identifying, I mean, we already talked about the data sets, right? Access to large amounts of data isn't necessarily the only thing. The problem quite often is it's fuzzy. The data is not necessarily high quality. And so identifying not necessarily just the holes in the data and filling those in, but also identifying which parts of that data might be wrong. And so some of the compounds that we had where we generated data on them, we realized that the data we generated wasn't accurate because, for example, the solubility is poor, and so the data that you're generating in some of the other assays may be unreliable. And when you do all these predictive tools, you can identify which are your outliers from an experimental data perspective as well, not just from a predictive perspective. And that was pretty useful data that we generated when we were working on our AI approaches to these programs.
Perfect. Thank you, Julian. Matt, that gets us to you. With Optibrium, you have worked with a range of organizations in different sizes, stages, and particularly with predictive modeling and generative methods. So what's your take on how does it compare to what Jeff, Darren, and Julian have shared?
Yeah, I mean, in terms of what I've seen and had the pleasure of working with lots of people across the industry, is that the biggest consistent area of success is definitely in predictive modeling, and as we've heard, has sort of become fairly standard, certainly in the larger companies. You know, we've had particular sort of successes bringing a new predictive modeling method to drug discovery. You know, we've heard Jeff gave a nice example that you know, most of these sort of new deep learning methods offer only incremental improvements over more classic machine learning methods. So we've been working at a slightly different way of modeling, which is deep learning applied to imputation of data. And this overcomes some of the really big challenges in drug discovery. You know, we've heard already about the sparsity of data. We simply can't measure all of the compounds that we're interested in all of the assays that might be of interest. And so how to actually make the most from the data that you do have, and what imputation does is to sort of fill in the blanks and highlight, you know, new opportunities and making more accurate predictions of that missing data. But it's not just about predicting properties. Julian just gave a nice example. By understanding and recognizing the patterns within that complex and noisy data, it can actually identify those artifacts and errors, the outliers that may represent, you know, an assay where there's been an impurity or poor solubility, and making sure that we don't get misled and miss opportunities due to inaccurate data. And then also answering the questions about, you know, if I can't generate all the data I might want to, what are the actual most valuable experiments to run? You know, what data, if I invest that time and effort, will actually give me the most information to make decisions? So we've been looking at sort of applications of this method across a range of different questions. And, you know, we've had a lot of success in a wide range of drug discovery applications in collaborations with large and small companies. I won't go through all the examples on this slide, you'll be glad to hear, but just to highlight a few of them, you know, thinking about combinations with generative chemistry. We had a great collaboration with the open source malaria project that was just published in JMed Chem just this year, to find new optimization strategies for an anti-malarial series and confidently identifying new active compounds. And then also we heard examples from Jeff about predicting some of these more complex and phenotypic screens. And so we demonstrated the ability to bring together sort of early data and more accurately predict those downstream expensive sort of cell-based or phenotypic assays that are just not tractable with conventional QSAR methods. And even taking it as far as predicting in vivo pharmacokinetics. So taking the sparse ADME data we generate early in a project even though that data is not complete, still being able to make, really state-of-the-art accuracy predictions downstream once we're looking at PK and animal models. And so this is where we've seen the greatest sort of consistent pattern of success, and it can make a real difference in drug discovery.
Perfect. Thank you, Matt. I think all of you have shared a number of successes. So there seems to be a lot of good things, despite some people now starting to say, actually, AI might be a lot of hype. But I think there's still a question, and I think a couple of you have addressed it already partly, but has AI really transformed drug discovery? So has it really changed the processes you are using for your drug discovery work? Or are many of the successes still more anecdotal in nature? Darren, maybe if you could kick us off and give us a clearer picture of that for GSK.
It's a difficult question because I think in some areas the answer might be yes, but we won't know for a number of years. So GSK's AI strategy has been heavily focused on biology and large genomic data sets and extracting insights and identifying new targets. So we won't know if that's transformed our work for a number of years. I think in terms of the areas I spoke about, we have been able to design in vivo active molecules in a small, very small number of design cycles in our pilot projects. And if you can then replicate that across a portfolio, then I think you're on their way to making a significant change to how we do our business. But we're not there yet, which I know we've got a third question around why that's difficult. Yeah, so I'll leave it there.
Perfect. Jeff, what does it look like in Genentech?
Similar. Other groups outside of small molecules and biology are investing a lot in generating new data to inform target discovery that will be interrogated with machine learning, which I'd call it's not so much a predictive problem, but pattern matching on a grand scale. What can you infer from all these different data modality types? As far as transforming drug discovery, I think I would argue the practice in the DMPK world already has. When I entered industry in the 80s and all the way through the early to mid 2000s, there were four main causes of attrition in the clinic. Business competition, like GSK is faster than we are, and we decide we're behind and we give up. That's still one. Another major one was safety, toxicity, unpredictable tox. Another one, lack of efficacy. And the other big one up until mid 2000s or so was DMPK, or poor PK/PD work. It took a long time, but the evolving practices in the DMPK world of developing better and better in vitro assays, establishing in vitro to in vivo correlations, eventually got us to the point where that's just not a significant cause of attrition anymore, because we can do those experiments. And now over the last 10, 15 years, we have pretty good predictive models. So we can diagnose and fix those problems while molecules are still in iterative medchem optimization. So I'd argue that's already transformed how small molecule drug discovery works. That's my favorite example to learn from, is I'd argue if we really want to keep moving the needle, we have to think about how do we attack the remaining sources of attrition, which remain primarily unpredictable biology, efficacy, and tox.
Perfect. Thank you, Jeff. Julian, what does it look like from your experience, and perhaps as well with light of your current organization?
Transform drug discovery? Yeah, absolutely. I think it's not necessarily, obviously the ADME side of things has definitely transformed what's been going on, and you have some clear timeline there to be able to see that. What's going on now is obviously, we'll judge that a little further down the line, as Darren said. But the other thing that's clear is actually the landscape of the companies. I think if you look across the organizational structures, or either the big companies, they've got a significant effort in the area, which clearly suggests that this was important enough to be able to invest time and effort and money into. But if you look across the small companies that are popping up all over the place or have been in existence for the last 5 to 10 years, there's a considerable number of companies out there that are focusing on this as part of their core principle, right? So AI or machine learning methods towards drug discovery, or the bioinformatics aspects, or protein predictions, structural understanding, which is exactly what we're involved in my new company, a New Equilibrium, trying to understand protein conformations of a tough to drug target family. So I think that's where maybe the next generation of stuff is, but has it transformed? Absolutely. I think just from the landscape of everything that you're seeing and people being involved, and I guess something else that Darren said, people in the normal drug discovery teams are looking for this to give them not necessarily a leg up, to make sure that they're falling behind their competitors. Because if you don't incorporate this at least some degree in your drug discovery projects, then you're falling behind and you're not taking advantage of a potential benefit.
That's definitely very great to hear that you would see it already as transformed in that degree, to that degree. Matt, what does it look like from your experience?
Yeah, so I've had the pleasure and the opportunity to speak to a wide range of key opinion leaders and in our customers and other pharma companies, and I think generally speaking that they're saying that some of these AI methods have demonstrated success in sort of one-off applications and proof of concept. I guess with the exception perhaps of predictive modeling, as we've heard, that they're not yet routinely applied and they're not sort of consistently being used across a broad range of projects. And at risk of treading on the topic of the next question around challenges, I think it's because it's one thing to apply these methods in the hands of sort of expert sort of data scientists and AI scientists, but it's another thing to actually deploy it to medicinal chemists for wider use. So, actually getting a robust, scalable deployment with sort of an intuitive way to work with these results that just makes sense in terms of the questions that med chemists and other discovery scientists are asking and the projects they're working on is a really big challenge. And also, as Julian mentioned, in the biotech space, they're not really fortunate to have big teams of data scientists to prepare data, to run algorithms and analyze the results. And they just need something to work off the shelf and just update as new data become available. And actually, even for big pharma, we heard about from Darren about the big effort that went into sort of updating the models. It can be very expensive and time consuming to do. And sort of to illustrate our approach to this, our Sorella platform, it's really designed to make this a sort of off-the-shelf, seamless process, sort of automatically updating and preparing the data for modeling and building new models as they become available. It's really important to work with the results based on the latest information and remove that burden and cost of manually updating the data, building the models, revalidating the models, and so on. And so we use a method called Alchemite. Jeff mentioned it before, which is this deep learning imputation method. And we talked about some of the applications. But then even once you've built and run the models, all of a sudden you've got a vast amount of data. You sort of fill in all of the missing data. You may have tens of billions of data points, and completely agree with Jeff's comment that in the drug discovery world, we really aren't in the regime of big data. But once you start to fill in all these blanks, you might have vast amounts of data to deal with, and then being able to very quickly search and access that data and proactively surface interesting results in a very intuitive way to the desktops of the decision makers is really important. And so I think there's a way to go. We have to have platforms like Sorella to be able to make this a sort of a day-to-day activity across pharma and biotech companies.
Perfect. Thank you, Matt. I think we had in parts already touched on that during your previous contributions. But what are some of the key challenges either you have overcome in the areas where drug discovery is transformed already, or where we are on the verge of hopefully seeing this transformation. So what are these key challenges we need to overcome? Julian, maybe we start with you. You were rather bullish already on the transformative potential of the transformation we have seen to date.
I mean, there's a transformation in the industry, right? You can see it as far as as you look around. But I mean, it comes back to what everyone says. There's clearly areas where it's been more successful than others, and some areas which still have some work to be done. I think what's remaining. One of the things that we're working on right now for a New Equilibrium, for example, is protein modeling, and I know that a lot of other companies are doing this as well. The problem there is, again, data. A lot of the data that people use to predict structures of proteins or motions of proteins, or all the force fields that are used in a lot of the foundational computational chemistry programs are built off of the data sets that are available, and the data sets that are available are based on structured proteins. And there's a lot of data there, and it's reasonably good, right? AlphaFold, etc., doing reasonably good jobs predicting the structures of structured proteins. But when you go to non-structured proteins or proteins with unstructured domains, that's still an untapped challenge. It's a problem that we need to solve to be able to drug these. These things are like 30% of the proteome, and they're involved in all sorts of important biology in terms of interacting with DNA, RNA, other proteins, all sorts of signaling mechanisms, cancer, cardiovascular diseases, neurodegenerative diseases. So being able to solve that, I think, is something that we should be doing, and that's what we're trying to do at New Equilibrium. I think the key there is that you're looking from a protein side, at least, at a closed set, right? It's not small molecule chemistry, it's protein chemistry. You've got 20 amino acids and all the potential post-translational modifications there. So if you break it down, it's actually potentially a closed data set. You generate a good high density, high quality data, quantum mechanics. You can train machine learning models around to understand quantum chemistry, and that's what we're trying to do there, to predict the unstructured protein structures and unveil this so that it opens up this area for drug discovery. So for me, I think that's one of the untapped areas, barriers and challenges that we should be able to overcome with AI and machine learning.
Perfect, thank you, Julian. Jeff, Julian has given us some of the scientific challenges. And I'm sure you have a number of those as well, but perhaps as well challenges when it comes to adoption and rollout. What would you see in that domain overall?
Right, so a large part of that actually was a bit of a cultural shift in getting, over a period of many years, medicinal chemists to accept models that were not perfect, right? These are probabilistic models, moving beyond very qualitative descriptions that came in the 90s, right? The Lipinski rules, right? Which provided very simple sort of rules of thumb, but now to a probabilistic model that says, hey, this design has a 80% probability of landing in a low clearance regime and a 30% probability of landing in high clearance, right? Rather than reporting an absolute number. We found that approach works pretty well for gaining acceptance, but it took quite a bit of time, I think, for medicinal chemists to buy into the concept that in vitro experiments can correlate well enough with in vivo outcomes to be useful, and that it's rather than worry about finding all of the molecules that could work, it's more about finding the ones that will not work. And to Julian's point, chemistry is really expensive and time consuming. I think one of the most important papers that's guided my thinking was in 2010, a really interesting analysis from people at Lilly and a few other companies breaking down all the parts of small molecule drug discovery. And when you look at their conclusions there, it comes down to about 50% of the total preclinical spend is in synthetic organic chemistry. That's med chem and process. And so that's informed a lot of my strategy computationally, whether it's physics-based or informatics-based or machine learning modeling. Anything we can do to reduce the amount of chemistry we have to do is going to be a win. But over time, I think in the companies I've been in and others we've seen, medicinal chemists are more and more enthusiastic about accepting sort of the reality that we can't predict new things very accurately. We're pretty good at predicting stuff that's unlikely to work. So I think I'll leave it there. There's some other things we'll get to later.
Then that gets us to Darren, just to see, Darren, how do you see it for GSK? I think, again, you shared some first thoughts on that earlier, but if you could circle back to that.
Yeah, I think in terms of challenges, one challenge is scale. So you see many companies when you know we're not alone. What we tend to do, we do pilot projects and we put the new technology in, and we really see nice focused effort, and we've got some good results. Now, the feature of a pilot project, of course, is that you heavily resource it. You pile in your expert people and your thinkers, and you're all over that project, giving it the best chance. So if you're now where we are, we've done the pilot projects, and you think, well, this sort of works, and you've got to now embed that across a portfolio. So now you cannot replicate that resourcing. You cannot replicate those people, because you've now got to actually, push this across. Your platform has to scale. Everything has to scale. And you've got to understand, do we have best practices or not? Where in a pilot, you're sort of finding your way. Now you've got to explain to people the best way to do this. So all of those things you have to now start developing. That's where we are. That's where I suspect many companies are. Is wow, how do we really now push this across and make it a more standard way of working? And so that involves technology, but it also involves a lot of people management and change management as well. So it is if you're going to get the best out of the methods, do have to change how you work a little bit. And to Jeff's example, med chemists have to be more accepting of some of these new technologies, and we can't spend 5 or 10 years getting them used to how to use a QSAR model, because this stuff is going so quickly. So yeah, but it's fun because the opportunity is there.
Perfect. Matt, again, you have worked with a number of organizations. So how does that match up with the experiences that Jeff, Darren, and Julian have shared?
Yeah, I think it chimes very, very nicely. It's about making these very sophisticated, cutting edge methods accessible to the sort of discovery scientists. It's great, as Darren said, to have your sort of expert team pile into a particular proof of concept project. But if you're going to put these in the hands of all the scientists, the experimental scientists, medicinal chemists, the biologists who are working on a project, it has to just work in a very natural and proactive way to help them to inform their decisions. And I think another thing, it's one thing to encourage people to adopt a technology, but I think particularly in the case of AI, there's a lot of suspicion about, well, is this just going to replace me? Am I going to be done out of a job by this technology? And I think absolutely it's not, in my opinion, about replacing scientists. It's about empowering scientists and helping them to do much more. And one of the things that we work very closely with our customers and with chemists and with other users is to make sure that we're actually addressing the challenges that matter to them in a way that makes sense to them. One example of that is our Inspira platform. And so this is about actually combining the expertise and knowledge of expert chemists with that sort of power that we've heard from Darren, particularly about AIs being able to rigorously do a tactical analysis of different optimization strategies, different compound ideas, but reducing that barrier. So it's not sort of going away and generating 100,000 compounds for you to sort of triage through, but constantly working in the background and optimizing compounds relevant to you with absolute sort of minimal setup. And then not just proposing ideas because the computer thinks they're cool, but actually learning from the expert chemist about what makes a suggestion relevant to them, and achieving this sort of seamless blend between expert chemistry knowledge and AI. And then very rigorously exploring lots of different optimization strategies, more than anyone could draw into a computer or even think of themselves. And so much more quickly getting to those optimal compounds and the sort of success stories you've just heard about. And so I think, yeah, reducing these barriers to access is absolutely key.
What would you then see kind of as the vision if asked, where is AI tomorrow? What does it do? Where will it go?
So yeah, just to sort of build on that theme, you know, we have come up with a term that we call augmented chemistry. So yeah, some of you will be familiar with the idea of augmented intelligence, which is this blend between human expertise and artificial intelligence. And there's been lots of success stories in areas like radiology and diagnostics and so on. And so it's really about going beyond software tools, a model that you run and then look at the results, for example, and in a much more sort of natural way, supporting chemistry analysis and decision making by taking into account far more data than anyone could have at their disposal or in front of them at any one time, and working in a much more proactive way to highlight interesting patterns, valuable conclusions, and you know, just working more intuitively to make better decisions more effectively. And yeah, I've given you a couple examples of that, but the key thing here is trying to avoid the hype, and work with methods that are sort of proven to work and demonstrated to have wide range of applicability. And so one of the things we focus on very much is publishing those results and actually sort of demonstrating where they work, and equally as importantly where they don't and where there's more work to do.
Darren, what would you say for GSK? Where do you see your next steps, and what does AI at GSK look like tomorrow?
I think if you look at the target discovery area, I think that they're getting close to. There's a phrase that you hear in machine learning circles of lab in the loop, which is very much about how do you actually use your algorithms like active learning to generate the data that you need to design the next experiment and go round. And I think that sort of concept is very powerful. It's almost an idealized computational science sort of perspective on drug discovery. But I think in some areas that's sort of where things are going. I think with the lead optimization thing, I think the opportunity with these ways of working is we can talk much more about optimization strategies. It's a very neglected topic in medicinal chemistry circles, but that's what we're doing, and actually making it easier to generate ideas and making it easier to use the models to identify what are the good ones. That then allows you to spend more time saying, and which is the best set to make next for this iteration? What's the set that we're going to learn most from, and to optimize faster? So I think that's sort of where we're heading. Hopefully these technologies sort of change the conversations we have, actually, and we really start talking about efficiency of optimization.
Perfect. Thank you, Darren. Jeff, what would you see going on in Genentech? So what are your goals for the more near-term future?
For the near term, it's focusing on some of those areas I mentioned on one slide, and what I'd say are some of the core areas: process chemistry, analytical purification. Anything we can do to make them faster will speed up getting to a candidate. Work going on in improving the way we analyze our HTS data, including phenotypic screening. And then I would agree with what Matt mentioned, and also Darren mentioned briefly, I think, a really cool paper from GSK called Bradshaw, which I think you named after John Bradshaw, right?
Very much.
Yeah, I knew him well, famous medicinal chemist, QSAR guy, cheminformatician. But I think that paper exemplifies what we would also call augmented science, not just chemistry, and that we would like our systems as we go forward to be to be able to analyze all of the data we've got and answer questions that an expert medicinal chemist like John Bradshaw would be able to do. We should be able to anticipate what are all the questions that we'd expect that expert scientist to do of software like StarDrop or Vortex or D360 or Data Warrior, whatever you've got, right? That provides the tools, if we can get the data into them, for expert users to analyze them and draw correlations. Most of those people won't look beyond a 2D analysis, right? We should be able to help them with tools like Matt mentioned or the Bradshaw paper, proactively answer questions we should expect them to ask, deliver those answers to them and say, what do you think? Are these good ideas that you haven't considered yet? There's a lot of software engineering required to make this happen in an automated way, but I think that's the vision for where we want to go. And we could bring to bear lots of tools there from cheminformatics, machine learning, and physics-based modeling. So that's kind of the high-level picture for where we're trying to aim.
Thank you, Jeff. Julian, that gets us to you. So what do you see in store for you and your organization?
I mean, right now we're recruiting. We're building a company and we're trying to do this. So obviously I'll be hiring a lot of people in this area. I mean, the one advantage I would say that we've got, and it's not across the board for all the small companies, is we've put computational chemistry, machine learning, augmented AI, etc., right at the forefront and the center of our company. So, and companies like Relay and others have done the same. I think when you do that, you can enable it in a small company environment because it's core and you can justify the expense. I think if you don't, then it's tough to really compete with the big pharma environments, right? Because you just can't afford to hire the whole team of people to do this. You can't afford to commit the finances, or you don't have access to all of the tools, right? You need to pick and choose. You've got like one computational platform, you're going to commit to it, and that's pretty much all you have access to, and you hope it's going to cover all of the needs that you have, because otherwise it's another 150 grand for the next one for the year for a different tool, or you need another person to hire in because they have the expertise that you need. So collaboration, again, I think is key to be able to do this stuff. But like I say, from our perspective, the key really is to grow the company and do this stuff. We're not in the right place to be applying the AI machine learning to our data sets ourselves yet because we're too early, but that's definitely in the future plans. We'll be putting a lot of this stuff in place to be able to optimize our processes and move the chemistry, hopefully move the projects forward a little bit faster, or at least a bit smarter. Right? It's interesting that the augmented chemistry, I mean, this is prescient of the sci-fi authors like Asimov, etc. You know, for years and years they're looking at cyborgs and people that you know combine the intelligence or the foresight or the breadth of experience that you have from a person, but then you apply the machines and the automation and the just number crunching capabilities alongside. And I think Jeff mentioned this, and obviously it's a part of the core of the Optibrium platforms as well, is the proactive or passive placement of ideas or suggestions or data in front of people, because again, everyone's busy and you want to go and do this stuff and you want to actively go and look at the data sets, etc. But if it's presented to you and you don't need to spend so much time and effort looking for it, it makes life so much easier. And access to all those tools for all of the industry, I think, is going to be key going forwards.
Perfect, thank you, Julian. But then coming to at least one of the questions. So there's a question: how do we deal with data quality and uncertainty in the data? Maybe Darren, you kick us off with your view on that.
Yes, very good question. Of course, we make great efforts, you know, to really make sure that's as good as possible. It's very key. Actually, one of the big learnings we had with our DMPK rebuild, because of we used to have a decentralized organization. So we had multiple assay protocols across those endpoints. That's very difficult to model. As soon as you standardize on one and you generate data, all of a sudden, lo and behold, your models get better. And so, you have to have a focus on these things. You have to, you can't just generate data and then say, oh, right, now let's use machine learning. It has to be a joined up strategy.
Matt, I know you're doing a lot of work on that question as well. What would your view on that be?
Yeah, I mean, data quality is very, very important indeed. It's the old garbage in, garbage out is the adage there. I think there's actually another question relating to this around literature data, and I think just to address that at the same time, one needs to be very, very careful indeed using literature data, because the variety of protocols being used, and you can't just mash all the data together that is ostensibly for the same property. You need to really very carefully look at the exact protocol that was used and how comparable it is. But one of the sort of things I mentioned before, and I think Julian also mentioned, is that you can actually use these AI approaches to identify data quality problems. If you have, outliers that, based on all of the other information, look to be highly improbable, you can actually very quickly identify those problematic data points and retest them or eliminate them from your data set, so that you're not sort of leading yourself astray. So that's also actually another application of AI, is assessing that data quality.
Thank you. I think we are getting closer to the full hour of the webinar. Maybe one last quick one, maybe Jeff, we start with you. So one question was that a number of AI companies are having press releases where they are disclosing drug candidate nomination within a year. What is your take on that? How convinced are you by this really fast-paced discovery work?
Right. Yeah. Good question. So no doubt they're moving extraordinarily fast. I think I would agree with Derek Lowe's analysis. He writes, of course, the very influential and I think astonishingly good blog called In the Pipeline. That most of these are in what we would call fast followers or drug repurposing categories. Some of them are mining kind of a niche indication. It's unclear whether their requirements for nominating a molecule to go to the clinic would be in the same category as a GSK or a Merck or a Pfizer or Novartis or us. The big pharma are pretty conservative when we go to that. Small companies, which I've been into, have to place bets faster and bigger because you just don't have the time and you don't have a large portfolio. So I think it is clear in some of those cases they are moving faster than we're able to do it. It's unclear to me whether that's really due to their AI or just the fact that they're a small company, and that's life in a startup, right?
Mm-hmm. No, I think that's a very valid perspective there. I don't know, Darren, if you would comment on that, and how do you see that?
I think Jeff nailed it. I think you always have to look at the detail of these things. I think because it's quite a furore about one paper published around claiming a cellular active in 42 days, I think it was, and you have to go and look at the detail. How much data did you start from? What change to the known molecules have you actually made in order to decide am I impressed by this, regardless if you're a machine learner or not? But we're all seeing drug discovery and said, am I impressed by that example or not? It's all in the detail for me.
Maybe we use that as the closing words as well. I think that probably holds true for a lot of the AI work, that it's really often in the details in order to say, does it work? How much does it help? And so on and so forth. I think on that, I would like to thank all our panelists, Darren, Jeff, Julian, and Matt. I think it was a great discussion. And it seems there are indeed first great successes of AI. I think Julian went as far to say a lot of areas have been really transformed already by AI in drug discovery. I think there were as well some caveats around acceptance of AI by the human expert. And I think it became very clear in all the contributions that neither of the panelists saw that AI replaces the chemist, but rather that it is a tool which helps, which augments what discovery organizations are doing today. And I think then there's still something around how proactive can AI be. So how much is it really helping to guide projects in a beneficial direction? I think Jeff had it on his slides, really to work on reducing the attrition we see the further down we move things in the value chain. With that, again, thanks to all our panelists and to you in the audience for joining us today. And we hope to see you soon for future webinars.